Hello everybody, welcome to lecture 10 of our course. The learning objectives of this lecture are to define the difference between mono and polygenic diseases, describe the genomic causes, examine the capability of personalized medicine and current pitfalls in its implementation, and interpret the challenges in genomic data sharing in bioinformatics. During this lecture, we will overview the types of human diseases, we'll define the principles of personalized medicine, specifically how it could be used in mono and polygenic diseases, its present and future development, and finally we will discuss several aspects of the ethical, legal, and social implications of genomic medicine and bioinformatics. Our understanding of causes and pathogenesis of diseases is changing constantly with scientific progress in the past century. Despite how little was known in the 1960s at the time of DNA structure discovery, some scientists have postulated that diseases occur at the molecular level. One of the pioneers of the molecular model of diseases was Linus Pauling, a very famous biochemist, educator, and social activist. Dr. Pauling has been awarded the Nobel Prize twice, in 1954 in chemistry and 1962 the Nobel Peace Prize, making him the only person who has received two unshared Nobel Prizes and one of the only four people who received the Nobel Prize more than once. His prize in chemistry was awarded for the discovery of chemical bonds in molecules back in 1920s, 1940s, but he was also among the avid supporters of orthomolecular medicine, megavitamin therapy, dietary supplement, and taking large doses of vitamin C. The orthomolecular medicine is a form of the alternative medicine propagating the use of dietary supplements in maintenance of optimum environment in the body and health. In fact, the term orthomolecular medicine was introduced by Linus Pauling to mean the right molecule in the right amounts. Sounds very close to the personalized medicine definition that we will talk about later in this lecture. Jokes about health and medicine have been popular always, particularly because medicine is one of the few fields in which every person has some knowledge, or at least the majority of population believes that they do. Almost any stranger can give you an advice on a medical condition, too many times without knowing much about the pathology. As we well know, it takes a person many years to learn the basics of medicine and almost an entire life to learn the specifics of some conditions. In addition, no other mammal inflicts such big harm to his body as humans do by eating and overeating, drinking and doing other activities that are not quite natural for our lives. That may be the reason why only 1/4 of what people eat are keeping them alive and the rest 3/4 are making doctors busy. In 18th century, the French philosopher Voltaire joked about his contemporary fellow doctors that they know very little about the drugs prescribed to the bodies, that they know even less against the diseases of which they know nothing. So, since ancient time, the difference between health and disease was one of the most debated by doctors and patients. In reality, we're not always sure if the condition is a health or disease because our perception of health may be erroneous. A well-known fact is that population health is much better, less complaints in military zone actions or during the war. However, does it mean that the diseases are less prevalent in these conditions? The definition of disease, in addition to patient perception, may also have different perspectives among the healthcare professionals. Depending on the background, some healthcare professionals see only the clinical presentations of diseases, while others are focused primarily on the genetic or genomics aspects of health and diseases. The bioinformaticians usually don't see the patients behind the numbers that are being analyzed. Today, the above specialties are collaborating very strongly to diagnose some complicated cases, and it is probably too high of an expectation to have somebody with all of the above perspectives being a family physician. However, knowing the basis of each of the disciplines may help in addressing patients' concerns in the most effective way. The field of bioinformatics involves the use of computer algorithms and databases to study genes, genomes, and proteins and their association with diseases. We could use a standard reference to compare the gene expression against it, or we could use a healthy subject sequence as a reference to compare with a patient. Once we know the difference in genotype, we could map it to the respective chromosome location and identify a gene or a transcription factor that may be affected by our differences in sequences. If the sequence falls into a protein coding region, we could estimate the functional effect of a particular mutation. Using functional genomic modeling, we could run scenarios like what if. Finally, we could use the orthologs of the genes of interest to examine any experimental functions in vitro with subsequent sequencing as needed. Such experimental data would be able to give us additional insights into human diseases and conditions caused by genes of interest. What sounded like a joke or a sci-fi scenario several years ago became a reality in some parts of the world. For example, if you are familiar with the South Korean city of Songdo, also called the City of Future or the smartest cities on the planet, the administration has already implemented a mandatory health monitoring feature for its population. The city was built in the past 15 years with a huge investment of about 50 billion US dollars, and it is a home to a number of international organizations and large high-tech companies. Each citizen of Songdo, also called Incheon, is offered an implantable or skin adhesive monitor that is able to connect with the doctor's office for health update and even drug delivery options such as reminders or even automatic pump activation if one is installed. So the personalized medicine will eventually become a norm to public health, hopefully without too much inconvenience to the patients and without an abusive intrusion into our privacy. Personalized medicine, also called precision medicine, is a set of scientific and clinical activities to identify genomic patterns of diseases, apply such knowledge in individual treatment or stratification of patients based on their genomic profile. As a general omics definition, personalized medicine is focused on tailoring the medical intervention to individuals based on their genomic, molecular, proteomic, or other specific biomarkers. The most advanced application of personalized medicine is related to biomarkers discovery and use of such biomarkers in development of companion diagnostics. From the FDA and other regulatory agencies' points of view, the companion diagnostic is a biomarker test that is being co-developed with a drug to identify the group of responders to the proposed drug based on the knowledge of the molecular pathway and drug's pharmacokinetics in the body. If the drug claims to affect certain molecules or pathways, the companion diagnostic is mandatory for approval. In contrast, the complementary diagnostic is a biomarker existing or newly developed that may help in patient management and treatment strategies, such as, for example, to evaluate the effect of drug in long term. It is an indicative measure and it is not mandatory for a new drug development and approval process. In general terms, a biomarker is a measured indicator of some biological processes, state, or condition. It does not have to be genomic or genetic, and some of the tests have been used for a long time in clinical practice, may be called as biomarkers also. Genomic type of biomarkers is a nucleic acid sequence associated with a specific process, disease, response to treatment, or just a known molecular pathway. So from bench to bedside, the translational medicine discipline is focused on a more rapid and safe introduction of basic science discovery into the clinical practice. Because such discoveries are many times very personalized, translational medicine is very tightly related to the personalized medicine. We talked about some rare diseases in our past lectures. As you remember, the drug developers of rare diseases are incentivized by the regulatory agencies to stimulate their interest in treating rare diseases. In comparison with some widely spread diseases, pharmaceutical companies are trying to develop specific new drugs targeted to particular forms or stages of common diseases. You can see from this slide that over 80% of so-called personalized drugs are focused on oncology, different forms or stages of cancers, and predominantly with known effector molecules such as receptors or surface markers. If we zoom into the oncology type of personalized medicine drugs, we will see that only a small portion of them have specific type of cancers as their target, with 31 out of 54 being developed for multiple types, followed by 8, 7, and 6 specific drugs against breast cancer, leukemia, and non-small cell lung cancer respectively. The simple explanation is that these are the most prevalent types of cancers and have a high rate of reoccurrence. From the total list of drugs in the late stage of development, 31 drugs or 18% are marked as those that require a companion diagnostic test or have one, and 21 drugs or 13% may have one or are likely to require as it becomes available, but specific molecular details are unknown at this time. Such biomarkers are used in stratification of patients, and once the group of responders is identified, the biomarkers may become mandatory for prescription purpose. The large majority of late-stage development drugs do not have a companion or complementary diagnostic biomarker because some of them do not have a specific molecular mechanism in its action or are not personalized medicine, meaning that their mechanism of action is the same in general population. This may be the cytotoxic drugs that are used in chemotherapeutic cocktails. Overall, in 2014, out of 44 new drugs approved by the FDA, 9 were labeled as precision medicine drugs as classified by the Precision Medicine Coalition. This table shows a number of precision medicine drugs along with their biomarkers that are specific measures of the mechanism of action or have surface markers that attract the drugs or are specific cytochromes that are responsible for drug metabolism inside the body. This January, President Obama announced a $215 million personalized medicine initiative. Aside from the obvious desire to get the right drug to the right person at the right time and right dose, the reasons for an urgent need in precision medicine is because, according to some latest statistics, very few drugs work on an entire population. For example, from most popular drugs used by a huge number of population, only 1 in 4 to 1 in 25 patients benefit from the treatment. For such popular drugs as statins, the ratio reaches 1 in 50 patients with expected beneficial effects. The chances of getting no effect at best and to end up with a harmful effect are too high by any measures. Partially, such disproportion is due to the skewed statistical numbers obtained from clinical trials in which a certain ethnical or geographical group of population participated almost exclusively, and the second reason being that we all have different genomic profiles which may affect how the drug is metabolized. Physicians have been known to use the so-called N of 1 trial, in which the physician is prescribing a drug and monitoring the effect in a single patient. For example, such practice is frequently used in treatment of hypertension, specifically idiopathic and long-lasting. A drug is prescribed and the patient is recording the effects in a form that then may be reported to his or her physician. In turn, the physician is adjusting the dose or adding additional drug or replacing it with a different brand or generic formula. From the list of blockbuster drugs used in the US, the drugs are helpful to only a small fraction of people depicted in blue on this image. The red people are those that at best do not show a beneficial effect. However, as we know, any extraneous substance may be a poison, and in many cases, adverse events are observed in large populations taking the approved medication by prescription. So the genomic-based precision therapy is a mandatory condition to improve the population health and effectiveness of treatment of diseases. Aside from the imprecision medicine, there are several examples in which the genomic profile is extensively used in patient management, such as, for example, leukemia, where 70% of patients are already getting their combination of treatment from a list of 40 drugs based on their specific genomic profiles. At some point, we may get to the situation when many polygenic diseases will be treated with specific targeted drugs, considering the genomic profile of each patient, similar to the situation with the classical genetic or Mendelian diseases. The more successful our medicine will become in management of pregnancies and deliveries, the more likely is that we will get a wider spectrum of genetic diseases after birth. Usually, many genetic diseases are causing an interruption in embryonic development, and a relatively small number of such diseases are reaching the postpartum period. This is contrary to polygenic or multifactorial diseases, which are picking up from the childhood of a person and are increasing in numbers as we age. You're familiar with categories of genetic and genomic diseases. On this slide, the polygenic diseases are distinct from multifactorial diseases, primarily because in the latest we consider environmental factors and chronic conditions as causative, while polygenic diseases are primarily defined as those in which more than one gene is specifically responsible for the pathology. The distinction is quite conventional, as I think that without additional factors, as in multifactorial, the clinical presentations of polygenic or even monogenic diseases may not be so evident. Complex diseases are another type that is better defined as idiopathic. We know that the disease is present, but we cannot explain why and certainly are not sure how to manage them, ending up in symptomatic treatment of signs and clinical conditions. Complex and multifactorial diseases do not have a clear-cut pattern of inheritance, although some of the phenotypic manifestations may be caused by the germline transmitted mutations. Mutations are usually associated with diseases. They may be inherited from parents, called germline mutation, or acquired after birth, called somatic mutations. The difference among them from the bioinformatics point of view is that the germline mutations are present in all cells, while the somatic only in affected cells. In addition, the germline mutations do not increase in numbers, while somatic mutations accumulate during the organism's lifespan. We certainly realize that presence of a mutation does not equal disease. Some mutations have evolved as a defense response to environmental or infectious factors, and some are silent, and some may be just causing genetic diversity without phenotypic manifestation and actually disease. Transmission of monogenic mutations abide by the Mendelian laws, with easily calculated odds of having or not a mutation in offspring. In monogenic diseases, we have only dominant or recessive inheritance patterns. Regarding the polygenic diseases caused by mutations in more than one gene or locus, the odds are very difficult to estimate as the numbers of affected sequences increases. The inheritance of transmission patterns is quite complex, and the clinical presentation of affected organs and tissues may be quite diverse in dependence on the accompanying environmental factors such as lifestyle. The genetic pedigrees of mono and polygenic diseases also look quite differently. In monogenic diseases, we can easily identify the sources of the genetic mutation, while in polygenic diseases, sometimes the source may not be the first parental generation that caused this mutation. As a reminder, this image has some details on signs and symbols used in composing or reading a genetic pedigree. In addition to individuals' graphical representation, the information about their relationships has also some specific conventional depiction, and it is not always easy to draw such a relationship from family history alone. For example, the questions on paternity may be very sensitive in some cases, or even difficult to clarify without a genetic test. Finally, the generational presentation also has some conventions, such as order of birth in a family, or representing the generations in Roman numbers. Aside from mutations in specific genes or loci, SNPs may be associated with specific diseases, as we already know from the GWAS studies. Specific genotypes may define the odds of genetic risks depending on the number of alleles affected. However, with SNPs, the situation is always not so obvious because we don't know for sure about the causative effect of SNPs, in contrast to causative effects of mutations in genes. SNPs may alter the protein functions and, in combinations with environmental factors, may cause exacerbation of some diseases or increased odds. In many cases, presence or absence of SNPs cannot explain the clinical manifestations of diseases. Patients expect the scientific community to develop innovative omics-based tests to more reliably detect diseases and to predict their likelihood of responding to specific drugs. However, the translation of these new technologies into clinical laboratory tests that can help patients directly has happened more slowly than anticipated. The pressure on researchers to bring the innovative methods to the clinic resulted in several publicly debated cases. Following a recent case involving premature use of omics-based tests in cancer clinical trials at Duke University, the National Cancer Institute requested that the Institute of Medicine establishes a committee to recommend ways to strengthen omics-based test development and evaluation. Based on the principles set forth by the IOM report Evaluation on Translational Omics, Lessons Learned and the Path Forward, the National Cancer Institute developed a checklist with 30 criteria for determining the readiness of an omics test for use in guiding patients' care in clinical trials. The checklist will be used when reviewing proposals for NCI-sponsored clinical trials in which omic tests will guide care of patients. Among those criteria, the sound validity of data is crucial in passing the bright line toward clinics and evaluation of tests' clinical utility. In the last decade, in the post-human genome period, the definitions of pharmacogenomics and pharmacogenetics have drawn a lot of attention. Pharmacogenetics is a science that studies how the drug will act on a patient in dependence on his or her existing genetic profile, while pharmacogenomics is focused on how the drugs will affect expression of genes in an individual. Currently, from the regulatory point of view, there is no difference, considering that the initial distinction was quite conventional to begin with. In many cases, the terms are used interchangeable. Historically, people observed many differences in the responses to drugs and food since ancient times. Only in the recent century, the basis for such differences became understood from the pathogenesis point of view. It is worth to mention that from bioinformatics point of view, the distinction of pharmacogenetics and pharmacogenomics helped to develop a number of related tests and approaches in understanding the genetic and genomic factors of human diseases. Today, both terms are the basis of clustering of patients in human trials in dependence on their genomic profiles. Presence of genetic factors in many cases are not indicators of diseases, specifically in GWAS studies, where we cannot be sure about the causative effect. The French philosopher and mathematician Jules-Henri Poincaré described the spurious and causative correlations as building blocks of a house, where the collection of stones would not make a house, similar to a collection of facts may not constitute a science. We should always be aware of spurious correlations when discussing the genetic and genomic factors in human diseases, particularly when there is no specific basis or understanding of the pathogenesis. One of the examples of spurious correlation is the comparison of US spending on science versus suicide records. During a 10-year period from 1999 to 2009, both events followed the same trend line with a correlation of 0.99, less than 0.01 point difference between the two observations. So basically, a logical deduction would be that the more US is spending on science, space, and technologies, the more suicides by hanging, strangulations, and suffocation we should observe. The nonsense is obvious and the two facts are unrelated. Each of them is true and the data were collected retrospectively. Similarly, with large number of data in genomics, we should always consider additional statistical and experimental conditions to validate our findings and to curate the results. To understand better the disease, we need to be aware of spurious correlation. We certainly expect our results and conclusions to be valid, but the lack of effect is not always caused by the mutation in a gene or by an ineffective drug, specifically in clinical trials and when the drug are self-administered by subjects. So, we all recognize the variability in drug responses, such as presence or lack of efficacy or adverse events, with at least 30% of patients lacking the expected clinical outcomes. We certainly realize that one size does not fit all. Why that would be the case? In addition to the genomic variability, some of the most obvious confounding factors could be the compliance to the indicated regimen, misdiagnosis when the real manifestation is masked by a comorbidity, drug-drug interaction, specifically for a poorly understood chemical entity, could be a crucial factor. Doses could be sometimes miscalculated, or in some cases the patients may believe that an increase in dose would have a better effect. We are starting to realize that not only the outside temperature may affect the responses to drugs, but also the day of time when the drug is administered within the circadian rhythm, seasonal fluctuations being obvious in some infectious diseases and manifestations. However, the genomic and genetic factors are responsible for the majority of variations in drug responses, and the approved drugs should have the genomic profiles associated with effectiveness of drugs included in the drug labels. Finally, we cannot discount the unknown unknowns. Bioinformatics is a very powerful tool in uncovering such unknowns, and today we have a number of technological opportunities to use the genomic profiles in diagnostics and treatment of diseases. The majority of present bioinformatics methods and techniques are using the genomic information agnostically without prior understanding of the potential changes in profiles, and interpretation of such data should be done with caution in order to avoid biased and spurious conclusions. So this was the last lecture of our course. I would like to thank you for being diligent students and for your hard work in the course. Good luck in your clinical and scientific activities. If you have any questions, please feel free to email me your questions after the end of the course. I will be happy to address any issues that you may have. Thank you.